For anybody watching the recording, there's your vocab terms. You can pause it right there, but we are getting started with notes on DNA replication. So, all right, here we go. Why is that? Let's draw a quick picture here. Okay. Simple picture, a little stick people. One, two, three. Three things that occur that relate to DNA replication. But let's think about like what's happening. What's happening in the arrow labeled 1? Growth. Growth happens. Okay. What's happening in the arrow labeled 2? Well, no, broken bone is before the arrow labeled 2. What? Healing. Okay, healing. And what's happening in the arrow labeled 3? Well, no, he's healed after 2, but 3 is producing 2 special cells that I labeled as, do you know the male and female cell? So it's simple, male, female. So what's happening in 3? That's an egg cell and sperm cell. Reproduction. Good. There must be another stick figure person around. They're in a very serious relationship. Sexual reproduction, egg cell, sperm cell. It's not really part of class. All right. So growth, healing, and reproduction. Okay. So those 3 things have something in common. Each of them requires a cell to turn into 2 cells. Okay, cell division. But now we've learned that a cell has DNA on the inside, and that DNA controls the cell's activities. It stores genetic information. Okay, so this one cell, to either grow, to heal, or to reproduce, and it's going to make some new cells. And each one of those new cells needs to be an identical copy of this one, so it needs its own piece of DNA. So what does one cell with one piece of DNA have to do in order to make 2 identical cells? Reproduce what? Yeah, but that's the second step. That's this part. That's cell division, technically. Okay. So what has to happen before the cell can divide? It has to make more DNA. That's what we call DNA replication. If you want to make 2 identical cells from one cell, before the cell can divide, you have to have 2 identical pieces of DNA. Because think about, can this cell right here turn into those 2 cells? It doesn't have enough DNA, right? So if we wanted to explain why it has to happen, DNA replication has to occur before cell division. So both cells get a copy of DNA. That's the big picture. If you don't copy DNA, you can't grow. If you don't copy DNA, you can't heal from injuries. If you don't copy DNA, you can't reproduce. Okay, because all of those things require cells to divide. And that means, can you guess what topic we cover in the next unit? If this topic is DNA replication, what's the next unit going to be? No? If this topic is DNA replication, the next topic, cell division. Because first you have to copy the DNA, and we learn about that first. And then you talk about how cells divide. Now, mutations are going to be a part of this unit. But the next unit is called, I think it's going to be up here somewhere, cell division. Let's see, cell division and also genetics, right? And so, like, I never would teach cell division before I taught DNA replication, because in time it happens first. You have to copy the DNA, then you can do that. Okay, so that's the big picture. I think that's the most important thing to understand, and I also think it's one of the easier things to understand. I need to make 2 copies so I can make 2 cells. The next part is what is it, right? There's a term that we're going to use. We just wrote it down. It's called semi-conservative, but I want to try to use a picture to explain what it means. So here's a piece of DNA. This makes more sense if I unzip it, not unzip it, if I unwind it. I'm putting 6 base pairs right there, and I'm putting 6 because we're going to find out next week how DNA works on multiples of 3. It's just always is multiples of 3, and I'll explain why that is later on. I'll put a 5, and I'll put a 3, and I'll put a 3, and I'll put a 5, and I'll put A, T, A, G, A, C. You guys should be able to tell me the complementary sequence on the bottom half or the bottom strand of DNA based on that. So opposite or complementary from the A, we get a.
Okay.
No.
Oh, T.
T. I'm trying to. You got to remember the base pair rule. Remember that's going to be on the test or on the quiz. Opposite from the T, we get a. Opposite, and then keep going.
T. C. T. G.
So complementary to 5 prime A T A G A C 3 prime is 3 prime T A T G C T G 5 prime. Okay, so now we can see it. All we did was untwist it. But then what happens is, I pointed out to you guys, and I said it's important that this is weak hydrogen bonds, and that means that they can be broken, and that means that this one can go up here, and it can be 1, 2, 3, 4, 5, 6. Now it's 5 prime A T A G A C 3 prime, and then this one can go down here, and it can be T A T C T G 1, 2, 3, 4, 5, 6, 5, and 3. Okay, so we've separated them out. We've opened up the DNA molecule. We'll pull them apart by breaking the hydrogen bond. What do we do now? Put it back again. Seems like that'll be a waste, right? We wouldn't have made anything. The key here is when we call this semi-conservative replication. If we were to describe that, each strand is a template for a new strand. So then some enzymes, which I'll talk about in the next page, can come along and they can make DNA complementary to this. 1, 2, 3, 4, 5, 6. And this. 1, 2, 3, 4, 5, 6, right? And so we end up keeping half of it. So the blue is new DNA, right? So this is new, and this is new. It is made in the cell. But the red is old from the previous one. Well, if we follow the base pair rules, then we should be able to figure this out. Why don't I do this one right here? I'll do the top one. T, and then start right here across from the T. There you go.
T. A. G. A. C.
And we switch the 5 to the 3, remember? This will be 3 and 5, T, A, T, C, T, G. How do these 2 sequences compare? How do these 2 strands of DNA compare?
Because they can also.
Well, no, but how do they, like, if you compare this to this whole thing, right? So this whole thing, what are they? The same. They're the exact same. Because we use the base pair rules.
What can I put on that to make it feel better?
I mean, Vaseline?
Does that work?
Yeah, I bet so. I mean, it'll contaminate anything in there, but we got tons of
I'm just doing air.
I've got some, but I can't find it right now. Okay. All right, so these are the same. You don't know what the word template means? I don't know how common that term is. Maybe, like, I looked this up. I don't know all the different definitions for template. When I think about the word template, I think of a plastic art template. Okay, and that would be like this, right? And so it's a little piece of plastic and you can use it and you can use it to copy things. And they'll always be the exact same, the exact same, the exact same. And you just shade inside there, you fill it in or something like that. So if you haven't heard that word before, that's what the word template means. And so when we call this a template, it means we can use it to make this, and this is a template we can use to make that. And because we're following the base pairs, they're the exact same. Usually, not always. We haven't got that. Are we good with that? You all see how it keeps half of it? The red part? Okay. This is the most technical page, and that's okay, but we can at least try to understand a little bit about how it's working. And I've unzipped it, as you can see, because that's the first thing that happens. Okay, this is the replication fork. Can you all see how it's kind of like a fork? In case you see that term, here's the handle of the fork. It's a two-pronged fork like that, okay? So there's enzymes involved. There are dozens of them. I'm going to give you two, just two, and the names are very, very descriptive as to what they do. In blue, we have DNA helicase, right there. Okay, its name is based on the double helix because it spins around the double helix. And it breaks hydrogen bonds. It pulls things apart. It unzips DNA. So which way is it moving?
Opposite.
Well, left or right? Yeah, it's going to the left, right? Because it's unzipping, which means that's already unzipped, and so it's unzipping in this direction. It doesn't make anything, but it exposes the information, right? The order and the sequence of the bases, the A's, and C's, and T's are now exposed and open, and the cell can do stuff with them. And that's when the other enzyme comes along. It is called DNA polymerase. It adds bases to single-strand DNA. So floating inside the nucleus, we have all these little A's, T's, G's, and C. And this enzyme, its entire job is to work as a puzzle maker. It makes puzzles, it completes the puzzle. And this puzzle, maybe it has right here an A. Which piece will fit opposite on the A? The T. So this one is going to go right there. Well, it just did all of these, right? Because we're unzipping that way, it just did all of those. And so it is also moving in that direction, and it's copying DNA. There's a problem here, okay? The problem is, I'm going to use my numbers here. This is the 5' end, right? So what's this? So what's this? Three, 5, 5, 3. Now we have it in several different areas, okay? DNA polymerase can only copy from the 3' to the 5' direction. So it's moving in that direction. And that means that DNA helicase is leading it. So we're going to call this one the leading strand. Here's the problem. It's also doing it over here. Okay, this one, maybe it finishes the puzzle, and then there's a C right here. So it's going to add a G right there. Which way is it going? But it can't, right? Because this is the 5' end, right? And so it has to go in that direction. It would make sense if it went left. It would be easier if it went left, but it can't go left because it always goes 3' to 5'. It makes DNA 3' to 5'. It can't do it the other way because it's not, the enzymes only work on physical shape. That's all it can do. Well, that means it probably just copied maybe these two right here, right? What's going to copy that one then? Well, but we have to copy all of the DNA. And that means that we have a problem that you'll see in the animation. And I really can't draw a picture of it, but we're going to call this one the lagging strand right here. Okay, it is technically, it's going to unzip a little bit more, and then another DNA polymerase will attach and go that way until it bumps into this and fall off, and then do it a little bit more, and then unzip and do it a little bit more. It's just more complicated over here on the lagging strand. For now, that's all you really need to know. To finish up this page, which enzyme up here would be responsible for making the mistake that we call mutations? Yep. It makes mistakes. We call those mutation. You think it'd be possible if you were doing a puzzle? You ever do puzzles? You have, right? I mean, everybody's done a puzzle. You can take two pieces that really don't work together. You can force them together and say, I finished my puzzle, right? For instance, like let's say this was a C. Well, DNA polymerase is supposed to add a G, and maybe it adds an A, and it doesn't actually physically fit, but it forced it in there and made it fit. And then it moved on and it did the next one and that, so then it left that problem there. That changed the information, right? It is an unwanted, it is meaningless change in information. It's like erasing a word in a book and just tossing in some random letters in there, right? So that's a problem. We don't know why yet. We'll talk about why here in another week when we get to things. But that's a mutation. And that's important when we get to next week with like, why do mutations cause disease? Why did Rosalind Franklin get cancer and all that kind of stuff? But we're not quite there yet. We'll build up to it. That's just the origin of those things that we call mutations. So, all right, let's do some math. Are we good with this? You remember how the math question went on the quiz? On the rates? Terribly. Yeah, terrible. No, like I said, about 10%, maybe 15% people got it right. That means like 100 people got it wrong on the test, right? So that was bad. So let's think of another question of time, okay? I need to look at my numbers here because. All right. So if I zoom out, I'm gonna do a quick. I'm not gonna label anything. I'm just gonna do a quick drawing. If I zoom out on that thing that we just did. That's what we just drew, right? Where we labeled everything and put the enzymes on it. It actually is happening from two spots. We call this the replication bubble. It's getting bigger in that direction, and it's getting bigger in that direction. And we're copying the DNA right here, and we're copying the DNA right here. Okay? So that's how it's actually happening. That's happening with DNA polymerase. But if we think about numbers here, there are roughly this many. What number is that? 3 billion, good. 3 billion base pairs. From here on out, I would like to abbreviate that as BP in the human genome. That is the DNA in every cell, okay? It's a lot. Last class was able to make a piece of DNA that is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21 base pairs in length, right? They have a long way to go if they're going to get to 3 billion, because this is like one base pair, right? One AT base pair. DNA polymerase copies at a rate of, and I'm overestimating this a little bit to make the math easy. It's really closer to 700, but I want to make it easy, so let's round it up to 1,000. 1,000 bases per second copied by DNA polymerase. Okay? You understand those two numbers. One is an amount and one is a rate. This is exactly what we're doing in the quiz. I'm going to give you the rate and the amount. I'm going to ask you how long it would take. How long would it take one cell to copy all of its genome, all of its DNA, in one of our cells if there's 3 billion bases and it copies at a rate of 1,000 bases per second? And no, you don't need to calculate. How'd you get 1 million seconds?
Oh wait, no. Is it 3 minutes?
It's 3 million,
Yeah.
because it's just a division problem, right?
Yeah.
And so all you gotta do is divide by 1,000, cross off 3 zeros. And that means that it is 3 million seconds to copy. That's a lot of seconds. How many minutes is that? What do I divide by? Yep. Equals, how many hours is that? What do I divide by? Oh, no, that's 60. 833 hours. How many days is that? What do I divide by?
24.
That's 35 days for one cell to copy all of its DNA so that it can divide, so that we can heal from injury. Does that make sense?
Yeah.
It does?
No, because I feel like 35 days.
35 days to heal from a small cell?
Oh, no.
Right? Because we said that DNA replication has to happen before cells can divide so that we can grow, heal, and reproduce. And now I'm saying it takes 35 days to copy DNA before a cell can divide. And those two numbers don't make any sense. We can fix that really easily. Okay? If all we did was copy from right there, it would take 35 days. How many places are we copying from on the picture above? Nope. Two. Right there as well. So all of a sudden, it is 17 and a half days. But what if instead it looked like this? How many places are we copying from here? Nope. 10. Remember, you're copying from both sides of each of the bubble. So this one is expanding this way, and this way, and this way, and this way. So in this case, instead of taking 35 days, well, we're copying from 10 places, so it's going to be 3.5 days. And it really happens in thousands and thousands and thousands of places. And so it takes about 2 hours for a cell to copy its DNA. It still is a while, but then that cell can start the division process, we can start healing. So it is a little bit more complicated than this. I think the math is an interesting one there. This is the math that I'm going to have on the quiz right there. Okay, not that same number, but that same-ish problem. All right, let's finish up with this last one because it's a little different in prokaryotes and eukaryotes. Which one are we? Okay, good. So everything we've been talking about so far has been eukaryotes. Okay. The prokaryotes, they also have DNA. Eukaryotes in the nucleus. Where's DNA in prokaryotes?
Floating around.
Floating around in what?
Inside
Floating around in what inside the cell?
the cytoplasm.
That's it. The fluid that fills up the cell. So it is in the cytoplasm. DNA in prokaryotes, in bacteria, it's a little bit weird. It's different than ours. It forms a circle. And it's hard to draw double helix in a circle, but they have a single small circular chromosome. One of them. We don't have a single small circular chromosome. We have multiple linear chromosomes. How many? How many chromosomes are in an animal cell, skin cell? Have you guys ever heard this number? It's a really important number for this class. Inside one of our cells, how many pieces of DNA make up the whole human genome? Nobody knows. 23 pairs in humans. I would assume so. I mean, that's a pretty common number to teach in when you're teaching like lower school or middle school kind of life science. That means, why do I say 23 pairs?
Because
As we move forward in this, we're going to talk about 23 and 23, 23 and 23, 23 and 23. I got 23 from mom. Who else did I get 23 from? Yeah, so mom gave me one copy of chromosome 1, one copy of chromosome 2, one copy of chromosome 3. Dad gave me one copy of chromosome 1, one copy of chromosome 2, one copy of chromosome 3. So 23 pairs, which is going to equal 46 chromosomes. But that's only in humans. Chimpanzees have 48. They have this weird thing where, like, they have a different number. And every species can have a different number. There's, not every species has a different number, but lots of species do have different numbers. So there are differences. But with these two things, which one's going to copy and replicate faster? Yeah, because it's small, right? It's really small. So this is going to be faster DNA replication. And which one's going to divide faster? Yeah, because those two things match up, right? So faster DNA replication, and that leads to faster cell division. And I like to toss in some reviews on prokaryotes anytime I get a chance. So a lot of this stuff, like if you were looking at it like this, you would not be able to tell the difference in prokaryotes versus eukaryotes. But if you zoom out a little bit, you definitely can because of the differences. We have 23 individual pieces of DNA making up that 3 billion base pairs. So, all right, put your notes away. Let's see if we can learn or do a quick activity here. So in normal years when we're doing, let me tell people over here really quick. Ella, we're going to do our paper modeling activity right now. You can't do that clearly. So what I would like you to do is the assignment that I attached, okay, the POGIL on DNA structure and replication. In normal years, we do a significant amount of like coloring and taping and cutting and paper modeling kind of stuff. Clearly, we haven't been doing that. One, we're not doing lab notebooks this year for students. But I still do think this is a really beneficial one. And if you get to a certain point, don't have to do the assignment. If you don't work fast enough, then you do have to do the assignment. But if you work quickly, then you don't. And plus, there's an opportunity for extra credit if you guys work really quickly. So I have a copy of these. I'm gonna give you all one of this. 1, 2, 3, 5, 6, 7. All right, how many nucleotides are on this page? 8. How many base pairs can you make from 8 nucleotides? Nope. 4. So you're gonna make 4 base pairs, and you're gonna make your own little piece of DNA. And we're gonna color it because I feel like that activates a part of the brain that helps us remember certain little details. And we want to make sure we all match the same color because eventually we're going to attach them together. The colors will be the deoxyribose sugar, like here's what you have, right? The deoxyribose sugar right here can be blue. The phosphate can be red, and the nitrogen base right here is either A, G, C, or T. So that's pink, purple, green, or yellow. I don't care if your colors are exactly the same. If you find one purple and you can lightly shade it for pink and darkly shade it for purple, that's fine. It's more about like identifying special different parts of this and then attaching them in the same way. Then you cut it out. Okay? When you cut it out, you don't have to cut like into all the little corners right there. That's okay. But then you start building up your 4 base pairs. And there's a super common mistake. Most people will take this one that is cut out and is a guanine, and they will take this one that is cut out and is a cytosine. And they will say, oh, just like a puzzle, there it goes. Nope. Or not.
Oh, because wait, what?
I can't. I mean, it might make the puzzle, but that's not how DNA is put together. It does. But there's another part of DNA that I'm not making correct if I do it like this. DNA is a double-stranded, anti-parallel, and complementary polymer of nucleotide monomers. One of those words I'm not doing up here. Yeah. No. What's wrong with this as DNA? It was a vocab word today. It's a major part of describing the DNA molecule. We've heard all about it. There's nothing wrong with the deoxyribonucleic sugar. Yep.
It's not anti-parallel.
It's not anti-parallel. They're pointing in the same direction, and they're supposed to point in opposite directions. So then you say, okay, I'm going to point in opposite directions. I'm going to take this, I'm going to rotate it like that. Now the phosphates are in opposite directions, but now the puzzle doesn't fit, does it? So what can I do with this one once it's colored to make the phosphate up here, but the G's and C's still point together?
Make the
You have to flip it like that. So the colors are going to be on opposite sides. And that's okay. That's exactly what I would expect to happen. And then when you get 4 base pairs put together, then you attach yours to another person's. I have all of these boxes up here where you guys can get started and work with. What? Oh no, I've got pink, purple, green, and yellow for the A, B, C, and D. There. I don't have tape for all of these, so as soon as we get one put together, then I'll leave some tape next to that one, and you guys can use that. But make your 4 pieces first. Turn the lights on. Last. 1, 2, 21 base pairs for 7 people. Oh, sorry, 21. 3 base pairs per person. Whichever class gets the highest base pairs per person value, I will add extra credit in.